Hello everyone, and thank you very much for coming along to this webinar. We hope you very much enjoy it. I know the last people are just trickling in, but I think most people are here now. So as you've noticed, this webinar is jointly hosted by the Physiological Society and the British Neuroscience Association. We thank both of them very much for putting this on. We've got two fantastic speakers today, and after their talks there'll be time for what I'm sure will be a lively discussion. So our speakers, I'm sure, will be known to all of you, are Steve McMahon and David Bennett. Very brief introductions, so I'll let them speak much more than I will. Steve McMahon is Professor of Physiology at King's College London, where his research group studies the behaviour, electrophysiology and anatomy of the somatosensory systems, and in particular pain. And for those of you who are familiar with his work, you'll know that his group have led the field in our understanding of basic physiology of neuropathic pain in particular, and the development of potential therapeutic targets. David Bennett is Professor of Neurology and Neurobiology and an honorary consultant neurologist at Oxford, where he heads the neural injury group, who have an aim to understand the nervous system's response to injury in order to promote repair in the peripheral nervous system and to prevent neuropathic pain. And that dovetails with his clinical interest in peripheral neuropathy and pain management. So I think we're up for a fantastic discussion. Just some housekeeping notices before we really get going. Please submit any questions that you have through the Q&A button at the bottom of your screen. We will ask the questions to the speakers during the Q&A portion of the webinar at the end, but if you submit questions as we go along, we can look through them during the presentations. You've got the option to submit questions anonymously if you'd like to do so. If not, please include your institution at the beginning of your question so I can announce this with your name. We may not have time for all the questions, but we'll try to get through many as possible. You can also upvote the questions if you like other people's, and type your own answers or comments in to other attendees' questions. So hopefully we can get a chat going there. And please note finally that this webinar is being recorded and will be available to rewatch should you miss anything. So thank you all very much again, and I'm going to hand over to Steve.
Okay, well, thank you. Let me know if you can't hear me. And can we switch to my slides maybe? So when I was asked to participate in this webinar, let me share my screen with you. I thought about the title, The Pain of Missing Treatments, and I took this to mean that we would have a discussion about some of the reasons why maybe the pain field has not been as successful as it might have been in delivering better treatments for people in pain. And that's what I intend to do. And so to that end, we put together a session in which I will speak for 15 minutes or so, and I'll introduce you to, is there a problem in pain treatments that haven't been developed? How are we doing? I'll tell you a little bit about where the pain field is now, but the most part of my talk is this item 2, and that's to start to address the question as to why maybe it's been difficult in translating our knowledge and perhaps how we could do better in the future. And then I will segue to a session from Dave, who's going to address the same kind of topics from a clinical perspective. I think using, as an example, his and other people's studies on diabetic neuropathic patients to try and get at this point as to whether we could do better and what will be necessary for that. And then we've left it a little bit open-ended. If we've got time, we both tend to rabbit on a bit. If we've got time, we will come back at the end and maybe address a few other issues specifically around drug development and analgesia. And obviously, we're happy to speculate about what's going to happen next week and next year. So first of all then, what is this pain and what are these missing treatments? And I think there's good evidence that there is a problem, and that evidence very simply is that there's a lot of pain about. Our drugs for treating it are not very good, and we haven't had many new drugs in the last decade. And let me just emphasize those points with a bit more detail. So here's some epidemiological data. This one is about 10 years old, although there's a more recent one this year actually out. This one relates to Europeans. There are similar studies from other parts of the world. You can see that what this study did is it simply surveyed tens of thousands of people across multiple European countries. And actually the primary result, which is this second bullet point here, I am still slightly amazed at actually. So the finding of this study, and it's not just this study, multiple other studies come to a similar conclusion. The primary finding is that about 20% of adults suffer from chronic pain. And in this study, the definition of chronic pain was not trivial. It was pain that had persisted for at least 6 months and was bothersome. It occurred several times weekly. And when it did occur, it had to be of a moderate or more severe intensity, rated as more than 5 out of 10 on an analog scale. And obviously, there were many other socioeconomic burdens associated with all this chronic pain that were drawn out in this epidemiological study, if you're interested. But one I highlight on the right-hand side of the screen, and that is here, a large number of people, the majority of people, said that the medications that they were offered were inadequate for their pain. And to explore that second point a bit, I'll show you some more data from a review by Nana Finnerup, in this case, looking at the efficacy of drugs, different classes of drugs, to treat, in this case, neuropathic pain. I don't think the picture is so different for other types of pain. But what she plots here is for different classes of analgesic drug, shown down the left. She plots a blob representing the size of the database, and she plots it against the NNT, the number needed to treat. So this is the number of patients that have to receive that drug in order for one of them to get a clinical benefit, which is usually defined as a 30 or 50% improvement in their pain. And you can see it's not a great story, really. So the best of these drugs have an NNT of about 3, and the worst of them approaching 8. So in the middle here, you have to give 4 or 5 people a drug before one of them gets better. So immediately you can tell that 60, 80% of them are going to be dissatisfied on this particular drug. And those people may be further dissatisfied because they get side effects from the drug. And I won't dwell on it, but part of the problem has been that there have been very few drug registrations for novel analgesics, certainly novel classes of analgesic drugs in the last decade. I could say that the migraine field has done relatively well, actually. That has had several novel treatments registered, most recently the approval of CGRP antagonists for the treatment of migraine. But broadly, we're not developing many drugs. And that's surprising, perhaps, because of what I'm going to tell you next. And that's that when you ask, well, how is the pain field doing in terms of its understanding? I would argue that we're doing very well in our understanding about the pathophysiology of chronic pain. And again, I won't dwell at any length, but just to give you a flavor of where the field is, let me start with this diagram that you could get from almost any textbook, highlighting kind of the neurobiological pathways that mediate our responses to tissue injury. And the idea is we have large numbers of specialized nociceptors, about 2/3 of our peripheral nerves, that are only activated by tissue injury. They're normally quiescent. When they're active, they send information into the CNS, mainly through dorsal roots, some through cranial nerves, that activates a series of ascending systems, which in turn fan out to drive activity in quite a widely distributed number of areas in the cortex. And that whole ascending system is known to be strongly modified or modulated by descending controls originating in the brain, coming down to spinal levels and regulating the transmission of this information. So that's the sort of the textbook pathway. The question we're interested in today is, why does that pathway become, what happens to that pathway in chronic pain patients? Why do some patients develop chronic pain. And there are several reasons, obviously. I'm going to focus on these physiological ones. I think Dave will pick up on genetic influences, which are definitely important, haven't been studied to the same degree or have not been discovered in animals. So he'll tell you about some of the clinical data and genetics. But on pathophysiology, there are several processes that clearly might contribute to the emergence of chronic pain in these disease states. So one of them is sensitization of nociceptors. That's to say the nerve terminals, the nociceptive terminals, become more responsive to applied stimuli. So they signal activity now in the presence of very small stimuli, and they give exaggerated activity to suprathreshold stimuli. And that whole process is called peripheral sensitization. An analogous process, a second form of amplification of pain processing, is known to take place at least in the spinal cord, almost certainly at other levels of the neural axis, and that further distorts or disrupts the normal relationship between a stimulus and pain. And it leads to, for instance, or it can contribute to gentle movements, bearing of weight on, for instance, arthritic joints becoming painful. And if that wasn't enough, it's now clear that there are other pathologies that may be at play. So these descending regulatory pathways are usually in balance, a balance of inhibition and excitation. And we know from a large number of animal studies that that balance may be disrupted and the inhibitory influences may be lost, leaving a predominance of facilitatory influences. So all of these mechanisms have been defined initially in animals. And one question is, are they relevant, which I'll come to in a second, are they relevant to humans? A fourth pathology, while I'm at it, which has really emerged mainly from functional imaging studies in humans, is to recognize that the connections are the cortical connections that contribute to our subjective sensations of pain. They're also malleable, they're undergoing modifications in a number of chronically painful conditions, and that may too disturb the normal balance of what is perceived as painful. So actually, I mean, you could say that we're spoiled for choice. We're too successful. We've identified these multiple mechanisms. I don't think I've got time to tell you, but we do have a pretty good understanding of the molecular identity of many of the players participating in these pathologies. And yet, we haven't been able to turn that into more effective treatments, despite the fact that we understand a lot more. And one reason, an obvious reason, may be that there is too many of these things. We don't know which of these pathologies are operating in which pain states, and I think even more poignantly, in which individual patients with a particular disease. So any of these processes may contribute. And I think an important question that I know Dave is going to pick up is how easy or difficult it is to stratify patients to get some measure of what their individual underlying pathology is, on the basis that we might then be able to apply this, this knowledge that we have been collecting so avidly for more than a decade. Okay, so let me skip. You have to trust me that we know a lot about the mechanisms of these sensitizations. So let me turn to the last point I want to address for 5 minutes, and that's why then is this understanding? What's the problem with turning it into better therapies? And you'll see I identify several potential problems. One, asking whether the animals and the animal models are fit for purpose. And the second, whether what we've learned from animals translates to people. So on animal models, a few things to say. The first is we have many, many models. It's not difficult to think of ways in which you can activate a pain signaling system. Many of the early studies were done using relatively non-specific stimuli. So carrageenan is an extract of seaweed. If you inject it into a pore, the pore becomes very swollen and tender. It becomes sensitized for a day or two. What it's a model of is not clear. People don't usually get poisoned by carrageenan. Similarly, formalin is an irritant that has been widely used for good reasons, but it's not clear what it's a model of. So even when we have understanding from these models, it's then very difficult to know in which clinical setting they would be relevant. More recently, there are many models of disease have evolved, which I think has been a major advance for the field. We have very good models that have at least some, often very good, face validity. You take tumor cells that tend to spread to bone, you isolate them and you put them directly into a long bone in an animal and seal them up. They divide and the animal develops a metastatic lesion in bone, which and shows a lot of sensory changes, which are very similar to those seen in patients. So these models have face validity and they are changing, I think, pain research. The other aspect about animal models is the endpoints we use to decide whether an animal is experiencing more or less pain at some point. And these are, in the vast, vast majority of studies, they are of the type illustrated here. That is, we put an animal into a box, we shine a light on its paw, and we heat the paw up until it chooses to withdraw. And we take that withdrawal reflex as a measure of its discomfort. Or we do the same with a stiff nylon filament that we push against the skin to exert a particular force. When the force is unpleasant, the reasoning is the animal will withdraw. And ditto if we squeeze the paw in a quantitative way with pressure. But you'll notice that all of these measures are reflex measures. They are, they're not, what the animal can't tell us whether it's in pain, so we are inferring it from its behavior. And in this case, it's not a measure of any ongoing pain, it's a behavior triggered by a stimulus we're giving. And that's one problem. A second problem is that these reflexes are known in many cases to be organized at a spinal level, or sometimes involving just brainstem reflex pathways, and certainly not engaging the full repertoire of circuitry that is likely to be present and abnormally functioning in pain patients. And one word of warning, I wanted to share one insight from a paper published 2 years ago by Genentech. I think this is a stunning paper that, like many stunning papers, as far as I can see, has been completely ignored. You have to buy the proposition that in this paper, they have a genetic model of a mouse that is congenitally insensitive to pain. And I think that's a very reasonable starting point. And what you see is if you study a number of pain models in this mouse, the wild type mouse behaves as expected, whereas the knockout mouse goes to cutoff. It doesn't seem to respond to the painful stimulus at all. So that's true in an incisional model of pain, studied with Hargreaves, a thermal stimulus. It's true in a CFA model, a model of arthritis. And it's a bit complicated here, but it's true in a model of neuropathic pain where the measurement is cold sensitivity. But what I point out here is that in all these cases, when you use von Frey monofilaments to study the animals, there is no difference or minimal difference between the wild type and the knockout animal when you use von Frey stimulus. So the conclusion to me is very clear that that pain-free animals still respond to von Frey stimulation as do intact animals. This can't be a measure of pain, and it should be abandoned, I'd say. The last point I want to make is that at the moment, the gold standard, the thing we all seek, is a measure of spontaneous pain in laboratory animals. Playing very badly for me is a video of animals speeded up. They don't usually look like this. They've all got chips in them. They're in a cage. They're sitting on a plate which has a number of aerials to identify where the animals are. The animals are being video recorded 24 hours a day for weeks on end with infrared cameras. And we are able to analyze a whole series automatically of motor behaviors in this natural environment in this cage. And I don't know if you can see it, but these are night and day phases, day 1, day 2, day 3, etc. And you can see that the animals are much more active at night than in the daytime. And here's a superimposition of about 6 animals. In this lower panel, we see animals that have had a chronic pain injury. And I plot out one set of data here, which we're quite excited about. It's not finished yet, but this is data from animals with cancer-induced bone pain. They start off about the same. We do an operation on them, a sham or a real operation. Their behavior changes a bit. They recover. And from about day 7, when we know the bone cancer model becomes established, these 2 lines begin to diverge, and they stay divergent. So I'm hopeful that this measure of spontaneous pain in animals will allow us to perhaps to get a better endpoint for the study of animals, and one that will translate to humans more. Now, I am out of time, so before I segue, I was going to show you some data to ask whether these pathomechanisms of peripheral and central sensitization, abnormal descending processing, whether those can be seen in human patients. And I'm happy to come back to it in the discussion. The simple answer is yes, they can. Very convincingly, we have quantitative experimental techniques to detect the presence of those pathologies in humans. So we know that the basic pathophysiology that we've described in animals is valid, and we know that it can play out in the human nervous system. The challenge, which I'm hoping Dave is going to pick up now, is partly to identify which pathology is occurring when, and whether that can be exploited. So over to you, Dave.
Thank you very much, Mac. I'm just going to share my screen. So hopefully everyone can see that, and thanks, that was a wonderful introduction. And I'm really going to move on to now kind of how some of these concepts can apply in the clinical domain. And I'm going to use a common disorder as an example, and that's painful diabetic neuropathy, which I'm partly using that because we're currently in a world epidemic of diabetes, mainly due to obesity driving type 2 diabetes. So actually it's a very common cause of neuropathic pain in the community. And approximately 50% of people with diabetes will develop a peripheral neuropathy. And of those people that develop a neuropathy, 30 to 50% will be troubled greatly by chronic pain. So there is actually immediately a very interesting question there, which is why do some people develop neuropathic pain and others don't? Can we discover any predictive factors for that? And this issue that Steve has already raised in his talk about can we stratify patients in a way that could help us target treatment more effectively. And part of this theme will be identifying pain mechanisms within these patients. Just to give some look at the kind of principal clinical factors, painful diabetic neuropathy, the neuropathy particularly affects the longest nerves in the body. That's going to be particularly the sensory nerves to the feet and the hands. So actually the distribution in the top here in red is where patients feel painful diabetic neuropathy, where their pain perception is, and you can see it's in the hands and the feet. And there is a relationship between neuropathy severity and pain, but it's a kind of fairly broad relationship, and that is that the more severe the neuropathy, the more severe someone's pain. But there are patients that have a very severe neuropathy and no pain, and there are patients that have a very mild neuropathy and quite severe pain. So what are these factors that are modulating this response? That's one question. And a second is, of those patients in pain, do we see clinical pain as one unitary phenomenon, or actually should we be breaking it down and understanding mechanisms and stratifying these patients? And I would very much argue for the latter. And one of the advances that we've seen recently in the clinical field is that actually we should be studying in more detail the changes in sensory processing that happen in patients, and that allows us to identify subgroups of patients. So one example there that's been influential is the German Neuropathic Pain Consortium developed quantitative sensory testing as a means of giving a certain sensory stimulus, in this case a pressure stimulus to the base of the thumb, and asking people's pain perception. You can then plot that versus a large control group, either in the positive going upwards in a gain of function, or a loss of function in the negative going downwards. and build up a sensory profile across lots of different sensory modalities, such as warmth, cooling, brushing the skin, repeated mechanical stimuli. And actually what they find is broadly 3 groups of patients. There are patients where mainly there's a sensory loss, there's a loss of function across all sensory domains. There's another group of patients that are particularly sensitive to mechanical stimuli, mechanical hyperalgesia. So they're in that sensitized state Steve was previously talking about. And there are another group that particularly are sensitive to thermal stimuli. And actually later on in this talk, what you'll see is the argument I will make that actually depending on the sensory profile, that is reflective of underlying mechanisms and also can have some efficacy in predicting someone's treatment response. So again, taking some of the themes that have already been introduced is that one key mechanism is sensitization within the peripheral system, peripheral nervous system. And a kind of really nice example of that was a paper from Troels Jensen and Soren Sindrup, which was to say that they took a group of patients, many of them had painful diabetic neuropathy, some traumatic neuropathy, and they did a very elegant targeted local anesthetic block. And they looked at both the spontaneous pain and the evoked pain. And they found that when they had this effective local anesthetic block, so they're completely blocking all afferent input, there was a marked reduction over minutes in both spontaneous and evoked pain. And really what that is telling us is it's saying that this peripheral input is really needed to maintain a neuropathic pain state. It's not saying that the central nervous system is unimportant, and actually you'll see later I will show how the central nervous system is important, but aberrant afferent input is very important in maintaining neuropathic pain. And we touched on this earlier, which is to understand the mechanisms by which nociceptors respond to stimuli and how that might go wrong in chronic pain states. And in the top panel here, this is human skin stained for nociceptors. These are passing up into the epidermis and shown by these arrows. And what happens in diabetic neuropathy is these are becoming damaged as a consequence of the metabolic state, and they are degenerating and dying back from the epidermis. And the point that Steve was making briefly was that we understand a lot more about how nociceptors work. So when I started in the field, we knew that there were various stimuli, whether mechanical, thermal, or chemical, that could activate these neurons, but we didn't know how it happened. And the exciting thing in terms of molecular biology of the last 2 decades is we now understand a lot more about the ligand-gated ion channels that respond to those specific stimuli and resulting in an ionic flux and a generator potential. We know that that can be modulated by G protein coupled receptors, such as the prostanoid receptors. And furthermore, we know a lot more about the mechanics as to how a particular group of voltage-gated sodium channels that show relative selectivity for expression in nociceptors amplify that signal and create regenerative action potentials that transmit it towards the central nervous system. Like many systems, that needs to be under some control. So potassium channels are important brakes in excitability. And there are also important pacemaker channels that will determine the rate of firing. So what's exciting is we've filled in a lot of this black box as to how sensory neurons work. And many of these molecules are actually important targets for analgesic drug development. Importantly, what happens after nerve injury, and for instance, in the context of diabetes, is that the expression of many of these ion channels can change. Not only does the expression change, but also their trafficking may change. And in the case of diabetes, where you have reactive metabolic species, there's going to be post-translational mechanisms. And all of that are contributing towards hyper-excitability. Now, I'm going to briefly touch on some human genetics, because I think that has helped us in the way we think about how the sensitization can take place. And I'm firstly going to have a slight tangent from painful diabetic neuropathy, and that's to a Mendelian pain disorder called inherited erythromelalgia. And that refers essentially to painful red extremities. And this is one of my patients, and you can see the redness of her feet. And she's doing what a lot of these patients need to do, which is she's in pain almost all the time, which is made worse by warmth, as is the erythema, and she's standing in a cold bath. And that reduces the pain level. And we've now got much more to the basis of what's the genetic cause of this. And in fact, that patient that I just showed you has a gain of function mutation in one of those voltage-gated sodium channels that shows high expression within nociceptors. That's NAV 1.7. And this is a hotspot for mutations. And as we've discovered more about inherited erythromelalgia, there are actually now more than 20 mutations in different domains of this voltage-gated sodium channel that seem to be driving neuropathic pain in these patients. And Steve Waxman and Suleiman Dibhaj in particular have understood the pathophysiological basis of this, which is what these mutations are doing is that they're making the channel more likely to open. They're hyperpolarizing the voltage dependence of activation. And when expressed in DRG neurons, you can see this mutation here is markedly increasing action potential generation within nociceptors. So this continuous primary afferent barrage from nociceptors is causing this spontaneous pain. So can we take parallels from this very extreme human genetic example to painful diabetic neuropathy? And we did that by studying quite a large cohort of very carefully phenotyped patients with diabetic neuropathy. And as I've said, some patients actually never develop pain, painless, and some patients do develop troubling neuropathic pain. And we said, could variants in NAV 1.7, could they be acting as a risk factor? So this is clearly an interaction between a gene and an environment, but actually that did prove to be the case. So when we were looking for rare variants in NAV 1.7, they were much more common in the painful group than the painless group. And again, one of the advances that we now see when we're studying ion channels is we have much more structural data. And so, for instance, with Steve Tucker, we could develop a three-dimensional model of this ion channel NAV 1.7, and we could see where these mutations map to. And for instance, this M1852T is in a region that we think is very important in activation of this voltage-gated sodium channel. And that allows us to make predictions that we can study electrophysiologically. And so we went on to do that with patch clamp analysis. We can express either the wild type or the mutant channels. And we did find actually quite marked changes in inactivation. And in simple terms, there's a depolarizing shift in the voltage dependence of inactivation. So they're less likely to inactivate. And if you look at window currents, which you can think of as drivers of excitability, with these two mutations, the window currents are much greater. And this is leading to a hyperexcitable phenotype. What's interesting is that these, unlike patients with an inherited erythromelalgia, they're not born or they don't develop neuropathic pain at a very early age in life. They can tolerate this phenotype or this genetic change for most of their life. It's the interaction with the diabetes and the neuropathy that seems to trigger off this neuropathic pain state. And so now I kind of want to just talk about the plastic changes that will be happening in the central nervous system. And we've already heard the introduction of the concept of central sensitization. And as I said, I certainly don't want you to go away with the feeling that the brain is unimportant in this. Certainly we perceive pain in the brain. One concept that's been introduced is that the brain isn't just passively receiving sensory signals. In fact, there's a top-down control which regulates transmission of nociceptive information within the spinal cord. And I kind of quite like to think about this as good cop, bad cop, and that there is a pathway, particularly around opioids and noradrenaline, that can switch off a nociceptive transmission, but there's also a pathway in other states that can facilitate nociceptive transmission. And the reason I like this analogy is if you've got young kids and you've seen the Lego movie, you'll know that the good cop and the bad cop are one and the same person, and their head is just spinning round. And of course, that's like all of us, which is depending whether we're anxious, our level of attention, actually that can quite rapidly affect our perception of pain. And there's now increasing evidence in humans that this system is important in having a deterministic effect on the level of neuropathic pain. And I'm just going to talk about a key hub in this pathway, which is the periaqueductal gray. And with colleagues Irene Tracy and Andy Segadal, who really led this study in terms of functional brain imaging, we decided to take a very closely matched group of patients with painful and painless diabetic neuropathy. And they were matched pretty much for every way in terms of neuropathy severity, glycemic control, all these aspects, other than the fact that one group had pain and the other didn't. And then we looked and we did see changes in this descending pain modulatory system. So when we were studying the connectivity between the periaqueductal gray and other regions such as the anterior cingulate cortex and the nucleus accumbens, we found that the correlation of activity between the ventrolateral periaqueductal gray and these other regions, the strength of the correlation of that connectivity was greater in those patients with the higher spontaneous pain ratings. And so that was evidence that this may be facilitating, acting as a facilitative function in neuropathic pain. And it should be said, we're trying to think how we can get data, these very complex data sets where we're looking at lots of variables of patients in chronic pain to understand the relationship between them. So this is just an example where each one of these dots actually represents a patient, and we've broken down a very complex data set relating to sensory function and some of these clinical variables in these patients. Females are in orange, males are in turquoise. And these are patients with painful diabetic neuropathy, and there's a variation in the severity of that pain, and that partly relates to some of the facts I've already told you about. So the severity of the neuropathy relates to that, as does HbA1c, which is a measure of glucose control, as does anxiety. And we can see what was interesting was that the gearing, the relationship between anxiety and pain was stronger in females than it was in males. So this is just a new way that by using this kind of principal component analysis, we can start break down and understand the relationship between these variables. Just moving on, I'm not going to labor the point of treatment, but we've already introduced the idea that it's fairly blunt in the way that we choose patients for treatment at the moment. So generally we're using information from randomized controlled trials. This is just an example of a paper from Nana Finnerup that actually Mac has already referenced. And that's where they looked at the available treatments, and they concatenated lots of different trials to come up with an overall idea of effect size of the various treatments. And then they could rank the treatments, and we've already seen this data shown in a different way, and broken them down into first, second, and third line. And one of the issues here is often, this is based on controls, where you're, for instance, taking all patients with lots of different neuropathic pain states, and you may think, well, actually, shouldn't we be targeting these treatments to the patients that are most likely to respond? So rather than this very unselected process to treatment, which is really based on large, non-selected randomized controlled trials, could we have a stratified approach? And actually, I already said that quantum centric testing is one means that we can use to try and break down different subgroups. There's a very nice example of this from Trolls-Jensen, where they were studying a drug, oxcarbazepine, which is a blocker of voltage-gated sodium channels, and actually has pretty poor efficacy in non-selected trials. But they came up with a hypothesis, which is that in a particular subtype of quantum sensory testing profile, the irritable nociceptor, which essentially refers to mechanical and thermal hypersensitivity, which I showed you earlier on quantum sensory testing, that the underlying basis for that is hyperexcitability in nociceptors, and actually these would be most likely to respond to a sodium channel blocker. And in those with sensory loss or non-irritable group, that there would be less of this hyperexcitability and less of response to oxcarbazepine. So what they did is they took a group of patients, and then a priori they used quantum sensory testing and put them in different groups, and they ran trials of oxcarbazepine versus placebo. And the oxcarbazepine proved much more effective, as was their original hypothesis, in the irritable nociceptor group than the non-irritable nociceptor group. Again, showing that this would be one way of targeting treatments to those patients most likely to respond. And finally, on the topic of treatment, in very rare examples are we seeing personalized treatment. So one example was in patients with painful diabetic neuropathy. We found, when screening for mutations, we found a mutation in another voltage-gated sodium channel, NAV1.8. And what was interesting about this particular mutation is that it was in an exactly analogous position to a mutation previously described by Steve Waxman and Solomon Dibhaj in the ion channel, voltage-gated sodium channel, NAV1.7. And actually structural data here, where we've in fact mapped NAV1.7 and NAV1.8 on top of each other, you can see that this residue is in exactly the same position. And the reason that we were interested in that is that it had previously been shown by Solomon and Steve that actually this mutation is very sensitive to carbamazepine, and this patient proved to be responsive to carbamazepine. So we could then express the homologous mutation in NAV1.8, so as it were, we're jumping from one voltage-gated sodium channel to another using previous experience and structural information. And we could compare vehicle, and then we could treat DRG neurons expressing this mutant form of NAV 1.8 with carbamazepine, and we saw a significant effect of carbamazepine. And actually, this patient did respond to carbamazepine. So I don't think we're going to see that commonly in clinical practice. It's definitely not a quick kind of work program to generate this kind of data, but it does give a nice example that in certain genetic causes of painful neuropathy or neuropathic pain, that we may take a very personalized approach. So I'm just going to end this part just by talking about some of the challenges in analgesic drug development, and then I'm going to pass back to Steve. It's not for lack of targets. And before we raise this approach, the neurology of pain, you know, we made some great advances in the last 2 decades, and actually one of the problems is prioritizing amongst all these different molecular targets. We've heard about some of the issues with animal models, and certainly I think one of the positives is that we've seen, as discussed, some improvements in how we can undertake that animal modeling. There is, by its very nature, a subjective aspect to pain report, but there's a lot of work, and I'll speak about it briefly later, about trying to get more information on how we can improve patient-reported outcomes, particularly with outcomes that are important and relevant to the lives, everyday lives of patients. The placebo response, and Steve is going to talk about that in 1 second, is very interesting, but also a challenge for drug development. And we've talked about the heterogeneity of patient cohorts and how stratification may help. So I'm gonna pass back to Steve and mute myself. And Steve, tell me when you would like me to.
Yeah, I really just wanted to make a few points about the placebo response, because it clearly has such an impact on evaluation of therapies in the clinic. And it's a pretty high hurdle for many drug treatments to cross, actually. I mean, it's interesting that one of the recent developments in treating pain is non-pharmacological approaches. And one of the issues there is that really it's not a level playing field. They're really, I mean, for epidural stimulation for pain, there's no randomized control trials done in the world at all. One's being done now in London, but they haven't been done. So there's a lower bar for some treatments. Anyway, with drug treatments, here's some interesting data. It's from a little assessment, a review published a few years ago. And it's a quirky finding that the claim is that the placebo response may be increasing over time. So it's clearly a problem when your drug has to do better than placebo, but it's even more of a problem if the placebo is getting more and more profound. And in this paper, they discuss a number of options as to why the nature of switched on smart people, why people may have more of a placebo response. If we're spending more on them and evaluating them in drug trials, maybe that's a need. But what this paper illustrates is that not much sign that our efficacy is getting a lot better, but the placebo is catching up with the efficacy, meaning the number of drugs passing that test is reducing. And I thought I'd give you one example of just how potent the placebo response can be. And if you could have the next slide, Dave, actually, it's a slide from work by Irene Tracy, and you can see the title here. They're asking how much expectation affects pain reports, in this case, in healthy people. And the experiment is to continually apply painful stimuli throughout an experiment, and they design the painful stimuli to evoke a certain level of pain, here about 65 out of 100 on a visual analog scale. And then on the next intervention, Dave, they start an infusion of this very potent opioid. It wasn't a massive dose, I think, to be fair, but nonetheless, it's surprising that the opioid on its own has a pretty modest effect on the pain reports of these patients when they don't know that it's being infused. And in the next phase of the experiment, they tell the subject that it's obviously it's still going on, but they tell them that it's starting now. And you now see there's a much more substantial reduction in pain reporting. And then the final very clever thing, I think, is that they tell the patients that they're stopping the infusion, but they don't stop it at all. And the patient's pain ratings pop back up again. And again, I think there's a little bit more animation, Dave, if you do the next couple. So yeah, this is the true drug effect. And as I said, this dose of remifentanil, it's quite small. This is the placebo response that you have to overcome to prove your drug is better. And this one, I think this is what Irene calls, this is your typical chronic pain patient. This is someone who has a lot of negative expectation from failed trials, miserable experiences, ongoing pain. So in this case, the nocebo, the negative expectation can have a dramatic impact on the pain you're feeling. There's no reason why this should be different from that, really, other than the nocebo. So I think it illustrates one of the ongoing features that is a problem. And in fact, there have been several thoughts about, could we find a better way of doing trials that would overcome the placebo response? And I'm happy to chat about it, but to my mind, the answer is no at the moment. So that's what I wanted to say on placebo. I don't know, Dave, if there's anything you want to say to wrap up, or whether we should...
I'm just going to quickly end on a couple of the positives, which is to say that we are seeing improvements in the way we do kind of clinical translational pain research. So I think one definite kind of theme of the last 5 years is big data. So UK Biobank, Genomics England, big QST databases. This is allowing us to take huge amounts of human data, which can relate to behavior, clinical factors, brain imaging, genetics, and really trying to understand how this might relate to pain risk, pain severity, in a kind of hypothesis generating manner, and also to test some of the hypotheses that might have come out of preclinical research. So kind of we're seeing translational pathway from the both directions. Traditionally, we haven't had great human cellular models. DRG neurons are not easy to access in the human, and induced pluripotent stem cells, which are differentiated into nociceptors, are a fantastic resource where we can actually look at their morphology, but more important, their function using electrophysiology. And that can be very helpful as a kind of intermediate step in the translational pain pathway. I'm not going to labor translation. I'm not going to labor stratification. We've already discussed that. But again, that's having a big influence in the way we design some of our trials that enable us to stratify patients. We're also seeing kind of completely new treatment modalities. So I guess traditionally pain treatments were mainly around the ideas of small molecule analgesic drugs and psychological therapies. We're seeing some completely new ideas, such as around gene therapy. So certainly neuromuscular fields have been revolutionized. For instance, in treating familial amyloid neuropathy from mutant transthyretin, we now have antisense and shRNA treatments licensed for clinical use that are revolutionizing the lives of those patients. And I think people are beginning to think how they could transfer some of this therapy into treating neuropathic pain and chronic pain disorders. And again, I think neuromodulation, we're seeing quite significant advances in the way neuromodulation is delivered to the spinal cord and also to the brain, and how that may impact on pain treatment. We're seeing biomarker development, and we've discussed about some of the advances in brain imaging. And again, this can be very helpful in seeing a pharmacodynamic effect of your drug in a relatively small cohort of patients. Again, really as an intermediate step before you start launching into those very large and at times expensive clinical trials. And just to end on the point of clinical trials, there's a lot of work going into improvements in clinical trial design, thinking about patient-reported outcomes, thinking about whether you use, for instance, enrichment designs with randomized withdrawal, and lots of work between private and public partnerships, such as the ACTION Consortium in the US and IMI Pain within the EU. And I think that's just very helpful to have that live dialogue about how we can improve efficacy and design of clinical trials. So I will end on the positives, and I'm sure we'd be very happy to take any questions. Okay.
Wonderful. Thank you very, very much, Steve and Dave. That's absolutely fantastic and very, very interesting, comprehensive and thought-provoking. We've got a number of questions. So Dave, do you want to stop sharing your screen and then we can...
Hang on. Right, I'm stopped.
Perfect. Wonderful. So I guess I'll ask a question, and whichever of you feels fits can answer it. One very close to my heart, so I'm going to take chair's prerogative and ask it, from Laura Klinkhammer, which is, do you think eventually that a personalised approach might be developed utilising brain activation patterns, for example, in the shape of real-time neurofeedback? And can we learn anything from the fMRI that might tell us how we can personalise this?
Yeah, I mean, I guess I can start answering that. Interestingly, Charlie, you might be better to answer some of the technical aspects of brain imaging. I certainly think some of the issues around that is how well we can assess spontaneous pain in patients. So and the mechanisms for that are improving. I think some of the issues are often in the start of brain imaging, people were looking at response to evoked stimuli. And the issue there is often the response to evoked stimuli doesn't relate to the pain that's bothering the patient the most. Usually the pain that's often the pain that's bothering the patient the most is spontaneous pain. And that can be, my understanding is that can be quite hard to quantify in an individualized patient manner. So I think, having said that, I realized, and I'm often speaking to Irene Tracy about the advances in brain imaging. So I definitely think that's an interesting area of development. I think how well you can personalize it and what kind of control imaging data sets you would need, that could be quite complicated. But, clearly, I think actually speaks to some of the opportunities that I discussed earlier about things like UK Biobank, where you're actually getting very, very large imaging data sets on large groups of patients that can actually start to allow you to generate some hypotheses that you could then test in smaller cohorts. I think that's a kind of thing that would allow you to do that.
Maybe I could add that there's a great case actually. Mr. Carl Cooke was involved in an industrial accident, and some psychologist put him in a scanner and submitted to the court evidence that his brain activation pattern was different on his painful arm versus his non-painful arm. And the judge admitted it as evidence, and he was awarded, he was given an increased award, severance award for his injury. And it's important because actually you can look online now and you'll find American legal firms offering quantitative assessment of brain imaging. So there's a great review of that written by Karen Davis, which I do think is a nice balanced report. And it says what Dave says really just then, that yes, in the future, it's certainly we're entering the realms of possibility, but right now, no. Yeah, certainly with this guy, there was no attempt. In fact, it's interesting. The first objection was that, well, he wasn't a card-carrying pain tester, so couldn't give a good report. But no, the analysis was very poorly done, and it was on provoked pain anyway, and all of the other technical considerations. So I think my reading at the moment is that maybe, but not yet.
I think one interesting aspect is the sheer variance in human data. It's the same with quantitative sensory testing. Okay, any kind of pain perception response, even to a stimulus, there's huge variation within the population. So how you're going to find that your one subject is outwith of normal when you've got to deal with that degree of variance, I think is very tricky. I suppose it might be different if you had a before and after. So clearly, if you have some kind of baseline that's individualized, that helps with that problem. But taking one person and saying compared to a normal population, I think that will be a real challenge, because there's just what I find very interesting variance both in brain structure and pain perception in a general population.
Yeah, no, I think
maybe I could say an answer to another question that I see has come up. I do think a better approach actually would not at the moment to rely on brain imaging, but to rely on some of these other laboratory techniques to try and get at mechanism. I think they could tell you whether an individual, Dave showed you a nice experiment you could, in theory, do in lots of people. You could do a peripheral nerve block with local anesthetic and ask does this individual patient have a pain that depends upon some peripheral pathology? I would argue that probably gets you much closer to a personalized state than we're capable of doing with I accept fully the power of imaging, but I just don't think it's come to fruition yet in this context.
Yeah, no, I think it is worth pointing out those results you were showing were group level differences, which are definitely which are there, but are not that helpful for
Yeah,
absolutely.
Subjects as yet. But yeah,
I'm not saying it's not helpful. Brain imaging has given us fantastic insights.
Yeah, it does.
I think it's the issue of personalised to one person. That's the problem that I have.
I'm very interested in the data that you showed about the placebo effect, and I see some of our attendees, there's a couple of questions on the placebo. If I just take one of them from Paul Mullins, is part of the problem or placebo effect a result of the subjective nature of pain responses in human trials? And that is, if we are asking patients or participants to rate the pain, their responses may be driven by more than just the pain they experience. And social expectation and other factors have also been shown to impact it. Is there any improvements in objective measures of for a pain response?
Well, it's sort of the same question done in a different way.
Yeah.
As I say, the American courts have accepted that functional imaging can be a painometer. That, I think, is roundly rejected by the scientific community, including the imaging community at the moment. And we don't have, that is the problem. We don't have, it's a subjective sensation, and we don't have anything other than subjective reports to guide us about someone's pain experience. And what the questioner says is entirely true. I mean, it's clear that many factors, possibly what you had for breakfast yesterday, will influence the amount of pain that you feel. And that is one of the things that definitely makes it a very difficult problem. And one of the reasons why I think it is important that the standard of proof ultimately becomes a randomized controlled trial. I mean, not that that's without its problems, but it's, I think that has to be the only way forward to avoid too much snake oil and the like.
The placebo response is a fundamental neurological process, and really it's just tapping into that same descending pain modulation system that we spoke about. That is mediated by that same process, and people have said, can you somehow divorce the placebo effect from clinical trials? I don't think you can. I think it's a fundamental process. I do think that. So I think ultimately the proof of whether an analgesic is effective is, as Max says, is still going to need a randomized control trial. I do think, for instance, some of the group level responses with functional brain imaging can at least show you that a drug is impinging on those nociceptive pathways in a relatively small cohort, as I mentioned. So I don't think that is going to that's not going to ultimately replace. And in fact, Irene Tracy has some data that she can see those difference in cohort sizes where the actual pain report is not significantly different. And it's not saying that imaging supersedes pain report, because ultimately I think you're still going to need pain clinical trial. It's just that drug is engaging with those neural circuits and you're seeing a difference in response to that drug. And I think that's a helpful step. Personally, I think that's a helpful step when drug companies are trying to understand how their drug works and to proceed in clinical development.
Great. So slightly nepotistically, because I suspect you know George as well, George Tackley, who was at Oxford and is now at Cardiff, has asked something else about the placebo-nocebo response. He says, we know that the PAG-RVM circuitry is key to this response. Is there a way of switching this off pharmacologically for trials, e.g. with naloxone? And certainly for those of us who aren't pain experts, spelling out what RVM is might be helpful. I could do PAG, but not RVM.
It's rostroventral medulla, and it's the same circuit that Mac and I have showed, and we both showed it in different ways. So PAG down to RVM and then projecting down to spinal cord. I just think giving someone so I guess the trouble is it as kind of goes back to what I said earlier. It's a fundamental neurological mechanism. It just the placebo response just taps into that descending pain modulatory system that we know can modulate pain. I suppose it's quite interesting. Could you switch it off entirely? I mean, the trouble is, I think giving something like naloxone that's just going to block all endogenous opioid signaling. I don't think drug companies would be keen on having such a fundamental effect on the way the brain works in the trial. So So I don't think there is necessarily an easy way to switch it off. I mean, I agree with what he says about the neural circuitry, but I think it's not going to be something you can just switch off as part of a trial. I don't know what your thoughts are, Mac.
Yeah, I mean, a neat idea, could we circumvent it in some way? But actually, if you add the complication of another pharmacological treatment that may interact with the one you're testing, and in fact, don't forget that your drug could be operating on these pathways.
Yeah.
And so even if you could disable them, you then disable the effect of your test drug. So I just think that would drive a drug company, usually is a drug company doing the trials, it would drive them bonkers. And the whole business, it's unfortunately, it's not quite like the experiments we do on a day-to-day basis, where if we don't like something, we can go and run the experiment again in a slightly different way. We can try and, different dose, a different approach. The drug trials are phenomenally expensive. And, that's one of the problems I think they do fail, is that you're like a manic gambler who has to stake a very big bet on a single number, and you hope that that number comes up. And it doesn't often. That's the problem.
Yeah, I mean, that's a good point. And, serotonin and neuroadrenaline reuptake inhibitors, the whole theory as to how they work is on that descending modulatory system. So switching it off would not be good in
some context.
That context.
I think with the complexity of pain and the treatments and all the circuitry that's around, as a young researcher or people, individuals looking at going further into the pain research field, what sort of advice can you, as experts, provide our young generation of pain researchers?
Well, I'd encourage
it. I mean,
we need more researchers, yeah. And let me do a little bit of PR that it's obviously your researchers are interested in whatever they're interested in, and that's long may that continue. But when the filthy lucre comes into it, you may be interested in can you pursue your study in a way that is going to be fundable? And I mean, it is interesting. I mean, the MRC, you're sitting on the Neuroscience board, Dave, yeah? I mean, they are increasingly interested in understanding neuroscience that will translate. And so pain is an eminently practical subject. It's a real problem for patients. So I think it is intellectually fascinating. I think it's got a lot of practical value. And I do think it's quite fundable, actually. And I think all of those things are good reasons to work in the field. Yeah, come on.
I agree. I mean, for add my penny's worth to that, I would say embrace complexity. For me, that's what has always made it interesting. I mean, I've always been interested in chronic pain ever since Steve was giving me lectures as an undergrad guys in St Thomas's, because I liked the fact that this was a system that from the most peripheral terminals innervating the skin to complex circuitry within the brain was incredibly plastic, and it was and almost at every level of the neuroaxis, that plasticity could drive chronicity and pain, and I found that incredibly interesting, and I still do. And I think the other positive is whether you're talking about studying human subjects with brain imaging or electrophysiology, or whether you're talking about studying rodents with kind of complex circuitry tracing, live imaging. The techniques have really, revolutionized how we can prosecute pain research. So I would say it's ripe area to research because technology has moved on so fast and really impacted on the field. So yeah, I'm gonna echo your PR comments, Mac. Area to work in. It's highly clinically relevant. The governments both in Europe and US have cottoned on to the fact that we have a fifth of the population in chronic pain. We don't have enough treatments. We have a huge problem with opioid use. So actually, I would say on both sides of the Atlantic, funding has improved, quite rightly so, for pain research.